Porcine parvovirus infection is a common infection found in pigs worldwide. It is an important cause of infectious infertility in pigs and one of the organisms responsible for the Smeddy syndrome. The virus is widespread and difficult to eliminate. It is harmless to humans and other species and doesn't pose a food safety risk. However, it does have a significant economic impact due to low reproduction.
Etiology. Porcine parvovirus is caused by porcine parvovirus 1, also known as ungulate protoparvovirus 1. It is a category A infectious disease and a single-stranded DNA virus of the genus Parvovirus and family Parvoviridae. It is resistant to environmental degradation and disinfectants and can survive up to 4 months in the environment. It is remarkably heat resistant, surviving 65 degrees Celsius up to 30 minutes. It hemaggglutinates erythrocytes of guinea pigs and humans, and this is used in laboratory diagnosis.
Porcine parvovirus has got a worldwide distribution which include pig herds in South Africa. Due to the resistance of the virus, this virus can remain infectious for months in contaminated pens, facilities, and the environment. And this serves as a continuous source of new infections into the herd. The main sources of new infections into a herd would be contaminated facilities and pens where this virus is still persistent in the environment, as well as infected introduced animals into a closed herd. The transmission is mainly by means of oronasal infections, where infected animals, whether they are subclinical or clinical cases, shed the virus in their feces, urine, saliva, nasal secretions, and in the semen. Transplacental transmission to the fetus is possible, and the fluids in the fetal membranes is therefore a big source of contamination when the sows give birth. Infected boars can pass this on venereally to a sow, although they do still have a debate among researchers whether this is an environmental contamination of semen or whether it is an infection that can be passed on to the sow. Fomites can also be contaminated because of the resistance of the virus. They survive for a long time on these fomites, and rodents can also serve as a mechanical vector.
Porcine parvovirus has a predilection for actively dividing cells and requires cellular functions generated during the S phase of the mitotic cycle, the phase in which DNA replication occurs. The initial site of replication is the upper respiratory tract, and it then moves into intestinal lymphoid tissue, crypt cells, and other sites via the bloodstream. Reproduction losses occur due to the virus's ability to cross the placental barriers in pregnant sows and infect embryos and fetuses. Infection in early days of gestation, before day 35, results in fetus resorption. Infection between day 35 and day 70 of gestation results in fetal death followed by mummification. Infection after day 70 will result in the offspring being immunocompetent. The virus is apathogenic for non-pregnant adult pigs. If only a part of the litter is infected transplacentally, the litter mates will be infected by intrauterine spread of the virus, and so the same litter will develop all the changes described above.
Porcine parvo mainly manifests as reproductive failure in the dam. Clinical signs will depend on when the sow got infected during gestation. Infection early on will result in fetuses being resorbed and the sow returning to estrus. After 30 days, an infection will result in fetal death and subsequent mummification. Parvovirus does not infect all of the fetuses at once, but one after the other, leading to the mummified fetuses being of different sizes. Infection after 70 days will be met by fetuses able to mount an immune response. These fetuses will be born, but will be less viable and have characteristically dome-shaped heads and slow movements. Abortion is rarely associated with this disease. No other symptoms are seen in breeding sows and gilts, and no symptoms whatsoever are found in weaners and growers.
Parvovirus is non-pathogenic in pigs. This means no cross-lesion will be seen on dams. The most important lesion seen is mummified fetuses. If dams have parvo but do not abort, there is a chance that those surviving piglets will have developmental abnormalities. Parvo is described as somatic complex, which are stillbirth, mummification, embryonic death, and infertility.
This is what the different sized mummified fetuses can look like.
Polymerase chain reaction is the most useful technique for the detection of porcine parvovirus in fetal tissues, semen, and other samples. PCR is considered to possess higher diagnostic sensitivity and specificity than hemagglutination or virus isolation. PCR is therefore better suited for the detection of porcine parvovirus in otolized tissues. The detection of viral antigen in fetal tissues by immunofluorescence is also possible. The material submitted to the lab to perform the diagnostic should include the mummified fetuses and fetal remains. Serology is also possible when the fetal tissues are not available or the sample has otolized. A standard method used is the hemagglutination inhibition assay for the quantification of porcine parvovirus-specific antibodies. The results, however, may be affected by incubation temperature and the source of erythrocytes. In the presence of aborted, stillborn, or mummified fetuses. Fetuses older than 70 days of gestation. A positive hemagglutination inhibition titer in heart blood tissue extracts indicates an antibody response. In fetuses less than 70 days of gestation, with a crown rump length of less than 16 centimeters, high levels of viral antigen can be detected by hemagglutination inhibition in tissue extracts.
Unfortunately, there is no treatment for porcine parvovirus, so one would rather need to focus on prevention and control, and these aspects would then become really important.
Parvovirus is resistant and infective. Parvovirus-free farms remain uninfected only when high standards of hygiene are practiced. You must restrict the entry of new stock. To prevent reproductive failure on a pig farm, they must be vaccinated before they are bred. Vaccination is a more effective way to ensure gills develop active immunity before conception. Only inactivated vaccines are imported into South Africa. They should be administered several weeks before conception in order to provide immunity throughout the gestation. Dams that are seropositive provide protection to their piglets via colostral antibodies. Maternal antibodies usually decline gradually to undetectable levels at 20 weeks of age. However, it can last up to 9 months and therefore interfere with active immunity developing in young gilts responding to the vaccine. Active immunity develops in response to infection with the field strain or receiving a vaccine. Antibodies can potentially decrease the incidence of clinical disease, but infection and subsequent shedding of field virus still occurs. The virus is well established in the environment and therefore prevalent in the pig population. Commercial herds aim to have herd immunity against the virus. In the past, gilts were purposefully infected with the virus before their first breeding season. However, this method is found to be unreliable and dangerous as other pathogens spread throughout the population. Now it is preferable to vaccinate the breeding females. Antibodies have been shown to last anywhere from 4 to 13 months, therefore regular revaccination of breeding sows is required. It prevents transplacental infection and therefore prevents fetal loss. Gilts usually receive their first dose at the age of 170 to 180 days old, or 30 days before insemination. The second dose is usually administered 15 days later. Sows are boosted usually 15 days after each farrowing. Boars can be vaccinated as well as boosted yearly. Vaccines used in South Africa. The commercial vaccines available include Farasure B, Suvaxine PLEN-B, Parvoruvax, and Parvosuin MR. They are all combination vaccines that also cover immunity against erysipelas that causes diamond skin disease in pigs. Farasure and Suvaxine also protect against leptospirosis. To note that these are all inactivated vaccines and should be used in animals of at least 6 months of age. The dosage is 2 ml injected deep intramuscularly into the neck muscles behind the ears. The vaccination program targets all breeding animals in the herd, including the gilts, sows, and boars. As porcine parvovirus is known as the gilt disease, it is essential that gilts are vaccinated twice before they are inseminated for the first time. Vaccine-induced immunity is boosted in a stepwise manner, so that sows that have farrowed at least once clearly have higher antibody titers than unbred gilts. Therefore, in practice, signs of porcine parvovirus may be seen in cases in which vaccination of replacement gilts has not been carried out or in which there is vaccination failure. Common vaccination failures can be due to the use of out-of-date vaccines, inappropriate injection site, for example in the neck instead of at the base of the ear, storage problems, and poor vaccination technique. It may also be that some animals do not have the required immune response because of a compromised physiological or immune status or a nutrient deficiency.
Thank you so much for taking the time to listen to our presentation. We hope you have learned something about porcine parvovirus. Have a great day.